I know that our next speaker is impatient for this weekend to end. For this coming week, she'll travel to the annual National Conference of the Wildlife Society, where she will receive the Excellence in Wildlife Education Award, the only one they hand out each year. Merav has, for the past 16 years, taught a senior capstone course at UW, and she gets students to learn the full character of what it actually means to do research, to go out into the field, to interact with, in this case, chipmunks, and actually capture them, count them, measure them, the problems of how do you capture them, how do you release them, how do you do it with the least amount of harm, and what do you do with them once you've got them, and what kinds of questions can you ask? And it's been such an experience that the Wildlife Society has picked her as the national recipient of this award. Professor, should I give you her full name, Merav Ben-David, is an expert on carnivores and their interaction with their environment. She's researched megafauna, polar bears, brown bears, and wolves, but also smaller animals such as minks, skunks, and otters. Usually her research takes place at the interface of water and land, where animals move back and forth from one area to the other. She's been at the University of Wyoming for 16 years, but Merav Ben-David hails from Israel and comes to us via Africa and Alaska. After earning 2 degrees from Tel Aviv University, she ultimately found her way to the University of Fairbanks, where she earned her doctorate, and much of her research has been conducted in northern climates. In fact, most of the photographs I've seen of her in the field, she wears a very heavy winter coat. I think she came to Wyoming to get warm. It is important to study the impact of disasters, both natural and human-caused, on the world around them. You know, in Jackson, we know of the Yellowstone fires of 1988 and of the impact that caused and the studies that have been going on in the decades since then. Professor Ben-David has been a key figure in studying the impact of the Exxon Valdez oil spill, which happened in 1988, long-term. And in her area of focus has been on sea otters, and as she has studied these communities, she has learned an enormous amount about sea otter interaction, both with environment, but also, and more importantly, with each other. And that's what we're going to focus on today, and how that sociability, that social character of the otters impacts not only the otters themselves, but the environment around them. So without me giving you any more hints, let me introduce Professor Merav Ben-David.
Thanks, Paul, for the introduction, and all of you for being here while all the fun stuff is actually outside. I shouldn't be laughing, so I'm requesting that when you do ask me questions, make them serious and not funny. I have a broken rib. And if I say ow, it's not because anything you did, but because I made a wrong step. I broke my rib last Friday. So it's in a healing process, but it's still painful. Paul, you got it all wrong. It's not sea otters, but rather coastal river otters. Everybody getting my R's? I do have an accent, so if you don't understand a word, please stop me and I'll transspell it for you. But we're talking about coastal river otters, and actually the name river otters is misleading because the majority of animals, you are familiar with river otters from the Snake and Jackson Lake and Yellowstone Lake. Many of you, how many of you have ever seen a river otter in the wild? Wonderful animals to watch, right? It's the same species that we have here in Wyoming, but in fact, most of otters, and we like to call them North American otters, although I myself slip, because the majority of them, if you look at abundance and total numbers of North American otters, you will find them living in coastal environments, both in the Pacific coast, from San Francisco Bay all the way to the Aleutian Islands in the Pacific, and all the way from Newfoundland to Florida in the Atlantic. So in North America, if you did a census of otters, North American otters, the majority of them will be actually living in marine environments. Many of them, especially the ones that I study in Alaska, have never seen a river in their lives. They live and die in coastal environments. Okay, so the name river otter is a little misleading, and we try to avoid it if we can. The story I'm telling you today started in March 24th, 1989, with the Exxon Valdez oil spill. Coastal river otters were one of the injured resources from that devastating oil spill. That's when we started studying them in Alaska as a result of the Exxon Valdez oil spill. I am not going to tell you one thing about what we found related to the Exxon Valdez oil spill, but if you're curious, you can ask me later. While we were assessing the effects of the oil spill, though, we discovered that river otters in coastal environments have a very interesting social system. Some individuals were solitary and remained solitary throughout the year. But some were highly social. In fact, the largest group we ever observed had 21 individuals. So intuitively, this caused us to wonder why, and I'll try to run this video from here, why this variation in sociality? And this video was taken by a game trail camera, and it's moving because an otter is playing with it. Orientation, we are talking about coastal Alaska. Yes, Alaska is a big island floating in the Pacific. No, it's not. It's connected to the Yukon here. I don't know why all our maps show Alaska as an island, but we're dealing with a very, very small area in south central Alaska, the Prince William Sound, where the Exxon Valdez oil spill occurred. Actually, the Bly Reef, where the boat went aground, was here, and all this coastline was oiled. This is our study area. It's 4,200 square kilometers. It's humongous. And basically, it's composed of a lot of little islands. This is the main area where we studied. This is called Herring Bay right here in the center of our study area, which is right here on this map. So to study river otters, you have to capture them in a vast area, and then we implant them with radio transmitters, and we follow them with a little plane, because you can't cover an area this size without flying, which brings us back to Michael's talk about flying, except this is sea level, so it's no big deal. And following about 55 animals for up to 3 years, which was amazing, what we saw is that we have a gender bias or gender difference here in sociality. And most of the animals that were in the high categories of sociality, that spent about more than 20% of their time in social groups, were males, and most of the females were actually solitary. So this variation in sociality was derived from gender, actually, they don't have gender. Let's put it in right terms, sex. Okay? We have males and we have females. Otters don't have problems with whether they are males or females, or issues with gender. Okay, so sex. We have mostly females are solitary and males are social, which is opposite of most mammals. If you look at mammal socialities where we have divergent between the sexes in sociality, what you find is that mostly females are social and males are solitary. So this was really an interesting observation. Another interesting part of this phenomena was that occasionally females would join male groups, whereas males usually occurred in all male groups. So when a female was social, she occurred with males, she socialized with males, whereas males often just socialized with males. So this was a very interesting observation. Okay, We did that in the years after the Exxon Valdez oil spill, and they told me since I was an undergrad, if you have a good story, don't go out and collect more data. It will ruin your story. But my PhD student, Adi Barokas, also an Israeli, but this is a non-related issue. He came into my lab, and he is finishing his PhD now, and he said, well, I don't believe this telemetry data, because how often did you fly? You know, once a week, once every 2 weeks. I want to do a fine scale study and see if indeed the females are solitary. I don't believe you, which is nice when your student questions you. But anyway, he adopted a very nice new system that we call EncounterNet, or proximity tags. It's the same technology that we use in cell phones. When Michael calls me, I know it's Michael because my phone says Michael Dillon. There is individual recognition of numbers and so on and so forth. The frequency of his cell phone coded in my phone. And so this is the same technology. What we do is, instead of put radio transmitters on the animals, we glue on these tags onto their backs. The tags interrogate each other, the same technology as cell phones, but they also are data loggers, so all the interactions are logged into the data logger in the little tag. And then when the otters come to those latrine sites, and we'll talk about latrine sites a little more, we put these base stations, you see the little antenna, that we tie them to trees, and when any one animal that wearing a tag comes onto the site, the base unit downloads all information from the tag, and we get full records of all the interaction that this tag has seen over the course of a few months. And this is just showing you what we found. This is just one example. These are 6 otters in one location, and you see that these two individuals interact with each other a lot and stay together. This is, you know, hundreds of hours, okay? They do separate occasionally, but they found together for long periods of time. These two individuals are largely together. These two individuals are largely together, but sometimes all of them come together and form these large groups. So the large groups are accumulation of smaller groups. At the same time, the females that we instrumented had absolutely no interactions with any of the males or any other female, supporting the observation, the previous observations that in this system males are highly social and females are not. Whew. Okay, so why? Why this uncommon social organization? So we use genetics, and we still use the older types of genetics for this analysis. It's called microsatellite analysis, because we do a lot of non-invasive genetic sampling, and if you do genomics, you will amplify the DNA of all the organisms that were in the environment rather than the specific animals. And by using microsatellite DNA that are specific to river otters, we can identify individuals. And this is one of my colleagues, Pam Groves, who helped us develop the method. And what you see is that you can identify individuals from their genetic fingerprint. This is just showing you 5 of our otters in 2 loci, and we use 9 hypervariable loci. And what you see, what you see is that in this locus, these 3 individuals are heterozygotes for the same microsatellite combination. This one is a heterozygote, but for a different one, and this one is a homozygote. But then when you add another locus, you can identify or separate these that look identical here. And actually, with 9 hypervariable microsatellite nuclear DNA sites, we can identify millions of river otters, which we never have in real life, just because they're very variable. So using these genetic fingerprints, we evaluated the degree of sociality from the radio telemetry in 2 ways, just dynamic interactions or frequency of association, versus a coefficient of relatedness. This tells you these animals are highly related, these animals are not related very well to each other. And when we plotted these data, what do you see? Anybody sees any pattern here? Are animals that are more closely related occur together more often? No. There is no pattern, right? We also looked at how many animals whether animals that are social have more relatives in a population than animals that are non-social. And you see that very few otters have very close relatives, such as parents or siblings, and most of the population is unrelated, but there is no difference between social and non-social otters. So based on these two observations, we had to conclude that otter groups are not kin-based, they are not family groups. They are a random association of animals from the population. And not only that, but that being social doesn't increase the number of relatives in the population. So you don't have what we call inclusive fitness. What about direct fitness? Do you have more offspring if you're social versus solitary? So we looked at percent sociality, again from the telemetry, and number of offspring based on the genetics. And what you see is that there is no relationship. There is no effect of being social or non-social on number of offspring you will have in the population, or a number of offspring you will produce. So we had to conclude that sociality had no effect on direct fitness of river otters. So they are not social because it offers them any benefits in terms of passing their genes into the next generation. And this is, again, from our camera traps, a female with 3 little cubs. Adorable. Okay, so if it's not kin selection that is driving sociality in river otters in Alaska, maybe it's an antipredatory adaptation. And we need to look at the potential predators. There are orcas. In fact, I've seen an orca attack otters, unsuccessfully in this case. Occasionally, they are predated on by sea lions, and the young, those 3 little young you saw, can be taken by a bald eagle. These are the 3 main predators. Now, you can imagine that in an area of 4,200 square kilometers, where you have to follow the animals with little planes and hardly see them, the chances of seeing a predation event, like I was really lucky to see that attempt, we can't actually document predation. It's impossible. But we can make predictions based on predation risk theory. And what our predictions were that females with young should be more social to protect those little things from the bald eagles, the cute things that you saw earlier. And otters should be more social when these predators have nothing else to eat but otters. And this happens to be in the winter months. And if similar to other mammals, a bigger group offers more eyes to detect predators, then larger groups would be more comfortable foraging away from the shoreline that offers protection from at least these marine predators. So let's see what data shows relative to these predictions, and what you see is that, first of all, females are less, as I showed you before, females are less social than males. Males increase in sociality. The increase in male sociality happens in the summer months, not in the winter months, okay? And we also found a negative correlation between group size and distance from shore, but it was really a really weak correlation, and I think it's a statistical artifact, not real observation. So we had to conclude that sociality is not an anti-predator adaptation, and that conclusion leaves us with the question, why are some of them so highly social? So then we have to look at the food that they can consume. And we have basically two alternative food sources for river otters. The first one being what we call intertidal, subtidal fish. These are fish such as this rockfish, greenling, ronquil, gunnels. These are little fish. Some of them can be fairly big. This Irish lord can get pretty big. But they live in a nearshore environment. They usually find shelter in kelp and rocks and structures within the intertidal zone. But they also have a lot of spiny features on their bodies that make them a little bit less palatable to a predator. They're ubiquitous along the shoreline and quite abundant, but they are they don't occur in large schools, so they are distributed evenly also along the coastline. So that's one potential food source. The other one are these seasonally available large pelagic schooling fish, such as herring and sandlance and capelin and salmon, that come into the nearshore environment to spawn. They usually spend most of the winter in the deep ocean or in the Gulf of Alaska, and then they migrate into the nearshore environment to spawn. This is a school of capelin. This is a school of sandlance. What you see in these two images are how these fish protect themselves. Oh, before I continue, they come in very large schools. So these are very, very large spruce trees along the shoreline. Compare the size of these trees to the size of these. The biomass of these fish schools is huge. And they don't have spiny basically spines on them, and so they're highly easily accessible. And because they're migrating fish, they have a lot of fat on them. Okay, they're very fatty fish. So this is how the two strategies these fish try to protect themselves from predator. When they are attacked by a single predator, in this case it's a salmon attacking sandlance, they radically spread, and they turn around sideways, and the scales flash and confuse the predator. So that's what they do when they're attacked by a single predator. However, when they're attacked by multiple predators, their strategy is to clump together. And then some of them on the outside will get eaten, but the majority of them will survive. And the fish fight each other to get into the middle as much as they can. You guys have seen videos of humpback whales hunting these fish, right? Where they go under and they release bubbles and they turn around themselves so the bubbles come up in a net. The net looks to these fish like a bunch of predators, and so they clump together, and then the humpback whale comes up and just swallows the whole school, right? You've seen those videos. Yeah, so basically this is the strategy the fish take when they're attacked. In this case, these are puffins, and they are attacked by puffins. Okay, so let's see, what do the otters eat? The intertidal fish or these schooling pelagic fish? To look at the otter diet, we use another innovative technology, which is called stable isotope analysis. And basically what we assess with this technique is the ratio of stable isotopes. In this case, you see carbon and nitrogen. I'm not going to go into details about this technique. It's been the basis of my career, and I'll be more than happy to talk to you about that in detail later. But what you can create when you look at carbon and nitrogen isotopic signal is basically a food web. You can, and this is an example of a terrestrial food web and then a marine food web, and what you see is that they differ in the carbon signature. And as you go up trophic levels, you have an increase in the nitrogen signal. And so, for example, if I was going to study what this mink ate, and I would get a signal here, I would know that it was a combination of these two prey items, okay? So we did the same with the otters, and the first thing we did was characterize their prey. And here is what these intertidal fish look like. in terms of carbon and nitrogen, or del C13 we call it, and del N15. These are the schooling pelagic fish, so you see there is a distinct signature. We also looked at some freshwater fish, but those are just very small three-spine sticklebacks, so the otters don't even bother eating those. So you see we have a nice signal of intertidal fish and a nice signal of these schooling pelagic fish. What do the otters eat? From each otter that we trapped, we collected a hair sample, and we separated the hair to underfur, which the otters replace between May and August, and guard hair, which otters replace between August and November. It's very similar to how dogs and cats shed. They first blow out the underfur, and then they blow out the guard hair, and then they replace the guard hair and regrow the underfur. Otters can't really do that. They have to first shed the underfur and regrow it, and then shed the guard hair and regrow it, because they're diving, and the only way they keep warm in the water is by having a layer of air trapped in their fur. So that was a very nice system to study the changes in diet using fur samples. So these are, every dot here has the same signal here, and what you see is a shift from mostly intertidal fish to more pelagic fish. And this shift happens more in males than in females. If you look at it just, let's say, on the carbon, because they are highly correlated, the carbon-nitrogen in this system, what you see is that the solitary animals, okay, low sociality animals, it mostly have the signal of intertidal fish in their fur, and they don't change much from August to November, or May to November, whereas the high sociality animals have much more of this pelagic signal and significantly eat more pelagic fish when later in the season. Okay? So this gave us the idea that sociality influences the diet of otters. And the question was, okay, so these fish are easier to catch when multiple predators attack them, right? The school claps together. So does sociality really give otters an increase in foraging success on these pelagic fish? So we conducted some captive experiments. We had otters, 15 male river otters, in the Alaska Sea Life Center in Seward, where we did oiling experiments on them as part, as I said, of our Exxon Valdez oil spill studies. But while we had them in captivity, we did a lot of other studies on them before we released them back into Prince William Sound. And what we did with them, and they loved those experiments because they got to chase live fish, is we had a very large pool, a 90 square meter pool, where they could dive and fish or chase live fish. We offered a single otter in the pool, either a group of 5 salmon, pink salmon, or these rockfish and greenling and so on. We did build a fish habitat from PVC pipes, so these slow fish will have a place to hide from the otters. It's not fair to throw into a small pool fish exposed where their natural adaptation is to hide, and let an otter try and catch them. So the fish would hide in the PVC pipes, and the otters would jump in the pool, and these are single otters. We let them forage for 30 minutes, and we recorded how long it took them to catch the first fish. And you see that it was much harder to catch the first salmon out of the group of 5 than it was any of the 5 rockfish that were hiding in a fish habitat. Okay, so definitely if you're a single otter, you'll be much better off trying to get this intertidal fish. When we put this is the same bar as this one, and then we let 2 otters into the pool with a fast fish, 3 otters, and then 4 and more, and what you see is that it becomes much more effective to get these schooling fish as a group. The reason why we have a little uptick here is because once you have 5 pink salmon in a pool and 4 more otters in the pool, basically the water spills over. Too much chaos. So anyway, definitely as a solitary otter, you're better off. You'll gain more energy and spend less energy on a slow fish or an intertidal fish, but you can get really easily to these schooling pelagic fish if you collaborate. Then we went back into the field data and we said, okay, do we see a match between the increase in male sociality with the arrival of these schooling fish in the spawning season? And we see a very nice correlation, very tight, okay? So we also looked at how far we find social otters from these schools of fish, and this fish data we collaborated with the Alaska Department of Fish and Game. They do these fish surveys for fishing boats. And so we looked at their data, where the schooling fish were, and then the distance from our telemetry between the social and non-social otters. And what you see is, depending on what time window you look at, 48 hours, 36 hours, or so, within 18 hours, social otters were found closer to fish schools than non-social otters. Okay, so all this data suggested that basically sociality in river otters is driven by consumption of schooling pelagic fishes. But that doesn't tell us why males are more social than females, right? Why not females taking advantage of this high energy, great resources, especially when they have to raise young and lactate, etc. They have higher energy requirements. We don't know. After all these years, we really don't know, but we think we have a pretty solid hypothesis. What we think is that the fish, these schools, are only found in one location within 18-hour windows. They move around. Sometimes these fish schools move around 90 kilometers, 200 kilometers, and go spawn somewhere else. It's like they are spreading their eggs into different baskets. They don't want to spawn in one location. Male groups can pick up and move 90 kilometers away and feed on these schools. Females, however, at this time of year have these little pups that you saw in the video, and they can't pick up and move. They're tethered to a den where they have their pups. So they can't take advantage of these schooling fish. But if the female fails to reproduce, and sometimes they lose their pups, or if she skips a year in reproduction, then all she has to do is join the nearest male group and forage with them. And this is why we see more females in mixed gender groups than we see males in mixed. They just basically join the nearest male group. Okay, this is all really cool and nice, but it's kind of anecdotal, and who cares about river otter sociality, right? Well, I do, but why would you care about it? Why is it important? Well, it turns out that similar to many other fish predators, river otters transport marine-derived nutrients into the terrestrial system and can have a huge effect on the plant and microbial community in coastal areas. Just to show you the model, we have ocean atmosphere interacting to affect the abundance and distribution of these salmon, sandlance, capelin, and herring. All these fish predators, including bears, etc., basically transport nutrients, marine nutrients, into terrestrial latrine sites, and they have a large impact. And all ecosystem ecologists that ever heard me talk about this system laugh at me and say, okay, come on, compared to atmospheric deposition, how much nitrogen can otters bring into the terrestrial system? And I calculated it. So on average, an otter eats about a kilogram of fish. That's a lot. It's about 10% of their body mass. Can you imagine eating 10% of your body mass in fish every day for years? Anyway, that's about 40 grams of nitrogen. Otters excrete the majority. If you're not a pregnant female, you'll excrete, or lactating female, you'll excrete 99% of it, so close to 40 grams. If you have, the density of otter is 1 otter per 2.7 kilometers shoreline, 100 kilometers will receive every year 500 kilograms of nitrogen, which is not a lot. Not a lot. If you try to fertilize your garden with this level, you won't get much response. But otters do not just spread their excreta along the landscape. They have latrine sites. I told you we'll get back to those latrine sites. If they are 50 meters square in size and are about every 700 meters along the shoreline, otters will deposit 75 grams per meter square per year. This is becoming a little more important now. In Alaska, we have double the densities, so basically each meter square of latrine site gets 160 grams per year. So that's substantial. And if you look at atmospheric deposition in Alaska, the next door neighbors to latrines only get 0.1 to 0.2 grams. So yes, river otter latrine sites get huge amounts of nitrogen from otter activity, okay? So I showed you that we have double the densities. By the way, the 2.7 kilometer, one otter per 2.7 kilometer is what we see in the Green River in Wyoming. The studies we did in Wyoming in the Green River were about those densities. So also a lot of nutrient deposition along rivers in Wyoming is happening. I told you that the densities in Alaska are double that in Wyoming, and so you'd immediately assume that the number of otters makes a huge impact on nitrogen deposition. But it's not the whole story. The question is, why do otters even use latrines in the first place? And you may have seen this already in previous videos, but this is really what otters do. They do what we call the poop dance. And here is one otter pooping. And here is another one. You see they have this funny dance that they do. The question is why? Why are they using latrines? Why don't they go everywhere on the landscape? So there were 4 competing hypotheses: reproductive status, male-female communication, territoriality, if you are solitary and you want to protect your territory, or within group communication, or between group communications or within group communications. So to assess these 4 hypotheses, we use the same captive otters that I showed you the diving data or the fish capture data before, and we expose them to foreign, unknown male scent, an unknown female scent. We got the scats from zoos across the United States, so they were animals from, Oregon and Washington Aquarium, etc. They shipped us the samples, and we looked at how long the otters, these Alaska otters, smelled the new scent. And these were non-estrus females, of course. You can imagine what would have happened if those were estrus females, right? One-track mind. But these were non-estrus females, these were regular males, and you see that they were more interested in the other unknown males than they were in the females. And within the group, the dominant animals in the group spend more time investigating male scent than the subordinate animals in the group. So that tells us that these scent markings first of all, facilitate these smaller groups coming together and creating the huge groups that I showed you the video of, but also that they are intra-group communication. I'm the boss, you're not. Okay, so these are social communications. So it's not just the number of animals, but also the social interactions. Okay, so now we have to decide how many animals there are, and we do that, as I mentioned, with non-invasive genetic sampling. We first collect the feces, and we also set hair snails. These hair snails were developed by my previous students, John Depew, and they're basically regular snails that we modify and change them from locking to a paper clip. The otter goes through, the trap cinches around it, and then the paper clip opens and the otter runs away, but leaves us a lot of hair on the barbs. And then we go into the lab and we genotype them again the same way I showed you before to get relatedness. And you see that this is an animal that was caught on 2 different days or identified on 2 different days on 2 different latrine sites. We use these capture and recapture data to create capture histories. Basically, we had 9 occasions, and on 4 we caught this animal. We feed these capture histories into what we call capture-recapture models, and we get a population estimate. And here you see that our overall population estimate for that particular study area is 123 otters. This is the confidence intervals, and we did it for another area, and that was 62, and this was the confidence intervals. The really cool thing about this is non-invasive, and I don't have to catch the otter to get at these numbers. And to me, it's still amazing that I can get an abundance and survival estimate. This is survival estimate from poop. Technology. Just to show you that these numbers really match nicely with telemetry data. So for this population, we estimated 65 individuals with capture and population reconstruction. And survival for these, we estimated at 1 to 94%, which is what you get here. So amazing technology. And I can estimate the number of animals. And following these animals and looking at the use of latrines, we realized very quickly that the changes in abundance and sociality of otters creates a huge variation in what we call the hotspots. So this is the same herring bay, the same study area I showed you at the beginning. And what you see is that you have a lot of deposition in these sites here, and then no deposition. These are heavily used sites. They're not in the following year. So basically, otter fertilization is really dynamic on the landscape and really affects the plants. Or does it? Okay, well, let's see about that. We spent a long time studying the plants. So even though I'm in the zoology department, I have a lot of publications on plants, which is cool. And this is Aaron Rowe, who did this study. And what we found is, for example, that the canopy, the leaf area per basal area of trees on river otter latrine sites. So if you go to those spruce trees that grow on the coast, and you go to a tree on a latrine site, and you measure how many needles does it have relative to the size of the tree, the basal area is how big the trunk is, you have larger, more canopy, more needles on the trees that are growing on latrine sites compared to the same trees next door, which are non-latrine sites. This is a 2.5 time difference in how many needles these trees would have. And think about needles as carbon sequestration. Basically, trees build needles, and they use needles to photosynthesize because they capture carbon out of the atmosphere. We also found that these trees that grow on otter latrine sites have many, many more seeds. This is going from distance from the shore upland. And what you see is that on the coast and all the way to 100 meters away from the shore, trees that grow next to river otter latrines have much more seeds, produce much more seeds than the same trees just growing off latrine sites. And we know it's the otters' fertilization because we had that fecal deposition signal in terms of nitrogen in these seeds. Okay, so otters' fertilization enhances seed production. And this is really cool because we use fish fertilizer on our gardens, right? It's the best fertilizer is fish fertilizer as well. The otters are doing it naturally. They're fertilizing the forest by eating on these fish in the ocean and then defecating and urinating as part of their social communication on the landscape. They also change the community. There is less plant diversity on latrines, largely through shading. The trees take a lot of the nitrogen. They have more needles. They shed the understory, and there are less plants that grow underneath. So huge effects on the landscape. Okay, so we have autosociality, largely males, not kin selection, not antipredatory behaviors. It's all about eating those schooling pelagic fish and then fertilizing the landscape. What will happen to all this when the planet warms up? Will the river otter social organization change? This is just showing you the blob. I don't know how many of you have heard of the blob? Oh, you better go to National Geographic 2 issues ago. There are some parts of the oceans that warm up differently than others. The North Pacific and the Bering Sea are warming very fast. The Arctic Ocean is warming even faster. And this is called the blob. And the blob has huge impact on marine life, major die-offs of marine mammals and birds and jellyfish and all kinds of stuff. But anyway, we do see a huge increase in sea surface temperature in Alaska because of this blob that sits right across from Washington State. And we definitely see huge differences in fish in Alaska as well. So this figure shows you what happens to these fish, warm water specialists. These are cold water specialists. And you see increases over time with warm water fishes, okay, mostly flounders and pollock and stuff like that, that are increasing, cod. At the same time, cold water specialists are decreasing. And here are capelin, a major source of food for otters, social otters, and herring, another important fish for them. So otters do eat these flatfish that are increasing, but these are individual fish. They are the intertidal type. They are not this schooling pelagic fish that drive sociality. And just to show you again that these schooling pelagic fishes come really near the shore. This is basically sandlands spawning in sandy shorelines. They are a huge and important resources for marine mammals and birds and people. This is herring for bait, by the way. And these fish are declining in the system because of climate change. So when the planet warms up, social relationship of river otters will diminish because the driver of this social behavior, the sociality, is diminishing. And the fairy tale that I told you today basically is that the availability of this fish is what is influencing sociality. Otters use latrines for largely inter and intergroup communication. That this social behavior creates a flux of nutrients from sea to land that is very important for plants growing along the shoreline and affects productivity and community composition. And when the planet warms up, all this will disintegrate because otters will have no reason to be social anymore. And I want to leave you with one message. I want you to imagine that you are so lucky, every single one of you won a free cruise from Seward, Alaska, right here, down to Seattle. And you're going to go on the Holland America or whatever, Princess Tours, and you're going to travel the Inside Passage. So congratulations, guys, you won. To get to Seward, you have to fly from Seattle to Anchorage. And if you're lucky, there will be no clouds, and it happens. Occasionally you do have this type of condition. That's how Google got this image. You will be flying along the coast all the way up. But the coastline doesn't look like this. This is really smooth, right? What you will see from the plane from 37,000 feet are a bunch of islands and bays and all sorts of nooks and crannies. and a lot of coastal rainforests. And you'll see them like this, and you'll see them like this. And I want you to keep in mind as you are flying over and enjoying this view, which is breathtaking, that on every single point of every single island, on every single head of land on that flight, 2,000-kilometer flight, there is a river otter latrine site. And we know that we emit a lot of CO2 into the atmosphere as well as other gases, and that globally these forests absorb about 30% of our emissions. So really having live, healthy forests is good for us. Logging is bad. It turns out that the coastal rainforests of British Columbia and Alaska capture about 10% of the total carbon sequestration of North America, which is a lot. It's actually, this is an underestimate because it was done before the bark beetle infestation. So it's probably much bigger now because a lot of our pine trees have died here, especially in Wyoming. And that these forests, when they're fertilized by river otters, the carbon sequestration of these trees increases by about 2.5 times. And so there's a lot going on that we can't see from 37,000 feet, but it is really crucial for our planet to persist as we know it. I'll take any questions. And while you ask me questions and I answer you, I would like to leave a slide acknowledging the huge contributions to this project by many colleagues, undergraduate students, lots of undergraduate students, graduate students, and of course, all the funding sources. So thank you. Yes?
I would be more interested in, I'm not sure what that word was, about the ocean blob.
The blob. Yes. So, okay, the ocean blob it's just a large area off the coast of basically Washington and Oregon, well into the ocean. It's huge. That has much higher temperatures than the rest of the Pacific Ocean. So just like the atmosphere is not warming up evenly across the globe, so are the ocean. It probably has to do with ocean currents and tides and so on and so forth. We don't know exactly what is driving it, but it's been there for a long time. And it is causing a lot of devastation to marine mammals and birds because primary productivity largely happens in cold water where you have upwellings, and when you have high sea surface temperatures, or even this one is pretty deep, primary productivity goes down. There are not a lot of phytoplankton, not a lot of zooplankton, not a lot of invertebrates, not a lot of fish, not a lot of fish, not a lot of mammals and birds. So yeah, there was a big article last issue or the issue before in National Geographic about the blob. Yes?
Cold water fish move north, presumably. Would not the otters follow and start to then fertilize other areas?
We have not yet seen a major shift in herring, capelin, and sandlins, but we do see some shifts north in salmon. Salmon are very adaptable, and they move, even though they show high fidelity to the river where they were spawned in or they emerged at, there is enough flexibility in their spawning behavior that they do move. It will be, and what we are seeing in Prince William Sound, actually, as the glaciers move back, we are losing glaciers, so tidewater glaciers are receding, and the fish start spawning there, and we see otters moving into these bays where the glacier used to be and not anymore. And what we see is a much rapid colonization of spruce, because the first plant that comes after a glacier recedes is alder, which is a nitrogen fixer, which is great, creates fertile ground for other species to move in. But we see that where the river otters have followed the fish and fertilized specific locations where the glaciers have receded, we see much faster colonization of spruce. So you can imagine that for spruce, especially coastal spruce such as Sitka spruce, not white spruce or black spruce, to reach, let's say, the Arctic Ocean will be a longer process. But within bays, where we do see glaciers receding, we see the otters and the plants and everything moves, yeah.
The second question would be what seems to be a difference in diet between the otters in California versus the otters in Alaska, and what impact that has on transfer of nitrogen?
Yeah, so where in California we have salmon, the otters are more social. But for example, in San Francisco Bay, I worked with a student who worked on otters in San Francisco Bay. The males are solitary and the females are social. And they mostly eat crayfish and not schooling fish there. And the females join together to protect the cubs from coyotes and feral dogs. And so river otters are really cool because they have this flexibility in social organization, and in response to environmental condition, they will shift their sociality patterns, which is really interesting. Yeah, so in California, the males are mostly solitary, and the females during the breeding seasons form this, we call them crashes. One female can stay with all the pups while the others are out foraging.
What's the percentages of males to females? Is it lopsided or is it 50-50?
Yes, very, very, very interesting question, because there are about 2 males per 1 female in otters when we trap them in the wild. And for a long time we thought that it's probably a behavioral bias. We're catching the males because they're more mobile, they move around a lot, they're less careful than the females. But we know from looking at otter carcasses that actually some of that sex bias actually occurs in uterus. There are more male offspring than female, or embryos than female embryo. Not quite 2 to 1, it's more like 1.5 to 1, but it then exacerbates later in life, probably because, at least in our system, and also in Yellowstone, we see cooperative foraging in otters in Yellowstone Lake because of, the cutthroat trout are basically salmon. And to catch those fish, we observed otters in Yellowstone form groups, not 18 individuals, but we have seen 5 otters in Yellowstone and so on. But yeah, the sex ratio is from basically fertilization, but exacerbates later, probably because males are more expendable evolutionarily. You only need 1 male, 1 good male, but yeah.
So how do they decide who they will mate with then?
I wish I knew. I don't, we know a lot less about the females than we do about the males. The females are, as I said, we catch less of them, and they're not interacting. So when we are trying to figure out what they're doing, if they come in contact with males, we've never seen it. I mean, obviously they do, right? But they're more difficult to study. What we do know is that when the males have these large social groups, come late March, early April, when it's the breeding season in Alaska, the most dominant males in a group. So the groups are multi-age. The female, these 3 little pups you saw, when they will be a little bigger, well, when they will be about a year old, they will leave mom. And if they're a female, they will emigrate a long way until they find a vacant home range. The males will just go they might not even leave their mother's territory, and they will join the nearest male group. So we see very different dispersal patterns between the males and females. And the younger males that are not reproductive remain in the home range and move around as a group. And the dominant, you remember there is a lot of dominance communication going on, those dominant males will actually leave the group and leave the area. And we had animals move 200 kilometers and mate with females far, far away, because most likely the female where they are was the mom. They move far, far away, they breed, they stay away for about a month, and then they come back to the same group and the same home range, and they rejoin the group. So we basically have this very fluid groups dynamics, but there is some core usually maintained by the younger males. And I have a couple slides back there, but I won't bother showing you that this is, we did network analysis and we showed that the young males maintain the group while the old males go mate with somebody else and then come back.
Well, thank you very much.
Okay, thank you.